My pleasure to turn to our first panel, and I will hand over to our wonderful moderator, Dr. Ruth O'Hara, our chair.
Oh, no problem. Yeah, we are going to have a series of panels today, and I'm going to talk a little bit more to the history of some of the work that we're now doing on sort of combining and blending models of cognition and neurocircuitry to best predict treatment response in mental health. And one thing I want to do is just cast a little bit the history. I believe sometimes understanding our history can be vital in making sure we take the right path forward. So if we go to the next slide, or can I do that? Yeah. I would if I'd brought my glasses. Let me see if I can do that here. Yeah, it's...
You got it.
No, there we go. Yeah. When I started my career, it was 40 years ago, I became a graduate student, almost 40 years ago to the day. And I was very interested in cognition, how people processed information. At that time, cognition in clinical terms was really only considered in the domain of aging, some of which I'm exhibiting very well here and trying to multitask, and also in the domain of ruling out for organic disorders. It'll tell you how long ago it was that actually neuroimaging was really only at the very beginning, and it was not routine to get a structural MRI. In fact, it was not at all routine, and there was no functional neuroimaging. So we often used cognitive tools to rule out for a distinction between psychiatric disorders and what were considered organic disorders. By definition, making sure that cognition was really not encompassed at all in the domain of mental health. Now, we've a vast amount of cognitive processes that this brain is capable of. And over the years, I started to consider, along with some colleagues, that maybe there was more interaction between your cognitive information processing, how quickly you could inhibit information, how you divided attention, what was your memory like, and so forth, and particularly those domains of what we now call cognitive control, executive function. This was augmented by experiences I started to have where I'd see actually cognition maybe more the source of difficulties that were being ascribed to mental health disorders. And one such issue of those sort of interface was I was dating at one point a gentleman who would suggest we'd go to a particular restaurant often for dinner where they never got a steak right. And I remember wondering why this individual would always suggest the same restaurant, even though when we'd go there, usually the steak was sent back and so forth. And if you were taking the psychoanalytic course I was taking at the time as a graduate student, you might interpret that as, in fact, an inability to overcome some steak that had been burnt in his childhood somewhere by his mother. But in fact, what I realized was my companion didn't have a very good episodic memory. I was studying memory at the time. And so I started to become the externalized episodic memory. Remember, the last time we went there, this is what happened. Oh, you're right, let's not go. And it really, that was one of many experiences that I had that made me think, are we ignoring a really fundamental domain by not thinking about cognition? I did go on to marry that person, but that's a whole different story. So we started in my lab, and there were others, thinking about cognition in as a source, or at least a co-occurring disorder with, if you had cognitive impairment, with mental health disorders. And we started looking at depression was the prime one. And I was very fortunate at that time. I was really integrated into geriatric psychiatry. And it was the one place where you could really talk about cognitive information processing as maybe a part of the profile if you had a late life depression, if you had a late life anxiety. We started doing more and more work on this. And parallel work was being done with schizophrenia, another domain where we really started to recognize in the 2000s, the early 2000s, the interaction between cognition and psychiatric symptoms. We found that they were associated with, indeed, poorer cognition was associated with depression and anxiety in late life. But what we started to see was actually a slightly different profile. While in fact memory dysfunction and executive dysfunction were key to depression, we found that actually inability to inhibit seemed to be a hallmark of anxiety. And that actually mapped onto some models of emotional inhibition that were being developed by Amit Etkin at the time that I started to focus on and actually persuaded him to come to Stanford too. We also started to realize this is not just a phenomena that occurs in older adults, it also occurs in younger adults. Nobody really does a cognitive profile assessment for individuals who are younger with depression, but that has started to occur, particularly in the last decade here, since 2010. And we've started to see, in fact, as well, the association of certain types of cognitive deficits with suicidal ideation. We really started to build a model here, and we started to deconstruct the emotional inhibition and suggest that, in fact, that's observed by deficits in working memory and information processing speed. So lots of work to be done. And it resulted in 2013 with Etkin and myself writing a paper that many of the neurocircuits that are involved in cognitive information processing, particularly in the prefrontal cortex, overlap with the neurocircuits that actually had been identified and starting to be identified as associated with certain mental health disorders. This soon became of interest to those predicting treatment response, starting again mostly in older adults. But Evangeline down at Washington University, now at Penn, did a beautiful paper where she looked at SSRI treatment response in late life depression and found that hippocampal volume, structural, was definitely a predictor of poorer response. If you were actually had a smaller hippocampus, that you had actually a less effective response to the SSRI. But she also found that an independent marker of treatment response was speed of information processing. And people who had already started to slow in that domain were also independently identified as poor treatment response remitters, so they did not really respond well. So we started to think that cognition could very broadly start to predict treatment response. But like so many things in our science, we discovered very quickly that this wasn't the case. We did a study that we looked at executive dysfunction in individuals with late life depression who in fact were undergoing cognitive behavioral therapy. And our prediction was 30% don't respond to CBT in late life, and 30% have executive dysfunction. These are one and the same. We found exactly the opposite. The individuals who improved most in their mood were actually those who had worse executive function. And we replicated that with problem-solving therapy. And what we thought was probably happening is the therapy itself was a scaffold for the cognitive impairments, particularly in organization and sequencing, that these individuals were suffering from. And so we are following up on that work. The story was much more complex when we looked at neuroimaging. We had actually fMRI pre-treatment with the CBT. We saw some patterns of treatment response, but sample size was small, very limited ability to actually see were there different patterns. And we saw too many different patterns predicting both response and lack of response to be able to make much sense of it. And this is one of the topics you're going to hear about today, where we'll actually talk about how do we use AI to capture that complexity that so far has not been something we can do. Leanne then brought an extraordinary vision to us. She said, look, we already have these wonderful circuits that have been identified, from the default network circuit through our salience networks. These may actually be circuits that already can give us more predictive value, particularly if we start to use them in interaction with cognitive measures. So, building upon this seminal work published in Lancet in 2016, it sort of gave us a more defined start and base. And really, some of the work that's come out of that has been tremendous, where Leanne has been absolutely a world leader in this work. Her and her team have started to do this more stratified prediction of treatment response. And in addition, in integrating this into our own models, we see that other disorders. Here is actually work that we did in 2019 on predicting treatment response from exposure therapy for individuals with PTSD. The neurocircuit is effective at predicting, but not near as effective as when you combine it with measures of memory function. So we're starting to get a sense that although we could establish a more streamlined base, we really are actually dealing with very complex information. The relationship of cognition and neurocircuits to predicting treatment response depends on the treatment. You have executive function associated with poorer response to biologic treatment like an SSRI, but better response to CBT. You have different populations where different cognitive measures are more involved. It was memory and not of executive function that was the best predictor for our patients with PTSD. But as our recognition of the vast amount of data, the vast number of neurocircuits, the vast number of cognitive measures, the vast number of treatments increases, how do we tackle this? I go back to feeling overwhelmed when Leanne said, no, we got this. So, the other thing I just want to mention here is that if cognition plays such a seminal role and is so integral to our treatment response, we can do things about cognition to improve it, and about emotional cognition, such as inhibition for emotionally valenced material. And so we've been doing work in this domain. But there are so many different modes to capture, so many dimensional approaches, and across the lifespan. What is the case when you're 20, may not be the case when you're 40, may not be the case when you're 90. And so really the model for going forward is to make sure that we're connecting these dots. And you'll hear on that shortly, that make sure we're bringing the multiple domains together. It was almost sort of considered odd to mention cognition way back in 1990 when one was trying to get it considered in the domain of mental health. To actually have the facilities that Leanne has outlined today. And then I think the new secret sauce is the state-of-art data science and AI approaches. As David Entwistle so eloquently spoke, this is a domain area where everyone knows that it may be the solution, but not a lot of people know how to use it or even to understand it. I feel in mental health, there's no excuse for that, because most of us have had very rigorous training in statistics of all kind and Bayesian models because of the need to have those in the absence of more objective data. We have a perfect marriage of data, of approaches, of tools, and of more objective measures from our new brain imaging, including externalizing of neurons via induced pluripotent stem cells. I think, Leanne, now is our moment, and I'm going to hand over to you to actually do the next talk. Leanne is going to talk about scaling and how important that is for reaching the next step. Thank you so much. We'll do that after. I'm going to sit down.
Thank you so much, Ruth. That was setting the stage of what have been some extraordinary developments in our field, and including the importance of cognition. So today, I will talk about, continue the theme of depression. And as we've highlighted, depression contributes more years to lost disability than all other illnesses combined. And as I mentioned in my opening remarks, it's a major cause of suicide. The WHO actually estimates that there's a suicide every 40 seconds. And as I shared, this is an awful statistic that fuels my personal sense of urgency for new solutions. Despite these really staggering statistics and incredible clinicians, depression and most mental health illnesses are still treated with a trial and error approach, trying one treatment at a time, seeing how it goes. So at our Stanford Center for Precision Mental Health, we're pioneering a new path using brain imaging and AI to define these circuit biotypes that can then be an objective way to personalize treatments. And I'll show you how we can double remission rates as a result. I've developed and patented a brain imaging processing system. You'll hear about the clinical translation of this pipeline through Atsiri later today. And here I show the 6 biotypes that Dr. O'Hara so clearly outlined that are now quantifiable through this pipeline. And they are the foundation for precision treatment. They enable us to move from this current one-size-fits-all to defining what is the root cause of your type of depression, and then matching it to the treatment that will be most effective for you. Each of these biotypes, the foundation of them is a key brain circuit. So let me introduce them. As Dr. O'Hara touched on, one of them is the default mode, and this is the one in blue that helps you reflect on your internal thoughts, your memories, and think about your future. The second one in green, the salient circuit, detects significance. So that could be internal states like pain or external changes in the environment. The yellow one, frontoparietal attention, helps us maintain our focus and sustain our attention. Then there are 3 that are to do with emotion and cognition. The negative affect circuit in orange activates with negative emotion, fear, and sadness. The purple circuit, positive affect, also known as reward, responds to immediate rewards like food, but also longer term ones like social connection. And the red circuit, cognitive control, is like our brain's executive. It helps us plan and it helps us suppress interfering information. These circuits are defining of the biotypes. 90% of the people we've looked at experiencing depression have a primary dysfunction in one of these circuits. And that enables us to determine what is their primary biotype. And our technology, similar to cardiology, as mentioned earlier, will use MRI to measure the brain and identify these circuits. So in our case, if I was, let's say I was in cardiology, I would use MRI to image the heart. I may do it under stress conditions as well as rest, and I would get at the root cause of very broad symptoms like chest pain and choose the right treatment for that cause. In our case, we're using functional MRI similarly to measure the brain at rest and during tasks to get at the root cause and engage these circuits. We acquire a functional MRI, and that scan gives us the information to quantify with the processing pipeline those circuits and to pinpoint your biotype and then match that to the treatment that's personalized to you. Let's remind ourselves before I go into some examples of the current situation. So with this trial and error situation we face, without having measurements, on average, only about 1/3 of people get better after multiple tries. And those multiple tries can be very prolonged, on average 7 years. So we really want to do better. And I highlight a few examples. This first example from the default mode is work led by Ji-Sung Ahn and Lara Folland-Ross. And we show that biotypes based on the default mode predict who achieves remission on different types of antidepressants, known as SSRIs or SNRIs. Individuals who we identify with high scores on default mode, the blue circuit, respond best to SSRIs, particularly sertraline. And when we match them, biotype to sertraline, we can improve their remission by 2.1 times. That's doubling the chance and getting closer to 70 compared to trial and error. By contrast, individuals have low function in this circuit do better on an SNRI that impacts norepinephrine as well as serotonin. And in that case, we can also improve their remission rates by 1.8 times. So this means biotypes can directly guide choice of treatment. I note that the ideal range is around 5. It's a Goldilocks principle, not too high and not too low. In the next example, we look at the orange circuit, the negative affect circuit. And here we're predicting between behavior therapy and antidepressants. Participants with the high scores do better on behavior therapy, improving remission by as much as 3.5 times. By contrast, those with low scores do better on antidepressants, around 1.4 times. And these insights help us match to the right treatment modality earlier. The behavior therapy we're studying here was developed by my collaborator, June Ma. It focuses on both activation and problem solving, and you'll hear more about her exciting work in the next panel. Going back to cognition, about 20% of people with depression have the cognitive biotype. This is with the impairments that Dr. O'Hara was highlighting. They don't respond well to standard antidepressants. They do respond well to behavior therapy. This cognitive biotype is marked by low activity in this circuit. We wondered what other medications might help. And after extensive searching, we landed on guanfacine. This is a selective alpha-2A receptor agonist, and it boosts brain plasticity, specifically in this prefrontal circuit. It's safe, it's FDA approved for other uses, and it's showing promise for this type of depression. We conducted a prospective biotype-guided trial, and that has just been accepted in Nature Mental Health, and it's through an extraordinary collaboration with the clinical lead, Laura Hack, and our investigators, Ruth O'Hara, Bawil Zhou, and Alan Schatzberg, and extraordinary, crucial early support from our supporters. We prospectively recruited individuals who met criteria for this biotype. Then we treated them for 6 to 8 weeks with guanfacine and rescanned them. And we saw some extraordinary outcomes. Clinically, after guanfacine, these individuals showed a 77% response rate. And all of those who responded, 85 actually achieved remission, which means their symptoms were restored back into the healthy range. And those clinical improvements were accompanied by a restoration of the brain circuit and also by improvements in their cognitive performance, which is how we function day to day. Here's one participant example from the first person in the trial. So as you can see, her scores are very low across all regions of the circuit. But after we treated her with guanfacine, she saw this remarkable improvement where her circuit function went back into the healthy range, her symptoms remitted, and her suicidal thoughts lifted. You'll hear more about biotyping with clinical cases in Eric van Staldoenen's presentation this afternoon. Now we're taking a deeper dive into the cognitive biotype through an NIH priority initiative called Impact MH. And I'm thrilled to be collaborating with June Ma and Olu Ajilore from the University of Illinois, Chicago. And Dr. Ajilore will be presenting his groundbreaking work in the next talk. We're also using AI machine learning approaches to validate the biotypes and ensure that they hold true across populations and data sets. And we're using AI further to refine the biotypes, to discover new ones, match them to many more treatments than we've currently studied, and hope to expand them to other mental conditions as well. And this afternoon, you'll hear about one of these exciting approaches using AI foundation models with imaging in a presentation from Dr. Teddy Akiki. So with the new flagship research space that we announced today, we're poised to innovate faster and reach many more people. And together, we'll make precision mental health a reality. Thank you.
So now we're going to be joined.
We're thrilled to introduce Dr. Olu Ajilore. He's a distinguished professor at the University of Illinois, Chicago, and a really important national advocate and advisor on our National Institute of Mental Health Council. Welcome.
Thank you for having me. And I really want to thank Leanne and the Precision Mental Health team for inviting me here to this wonderful symposium. It's really good to be back at Stanford. I spent a good part of my 20s here when I did my MD PhD. And it was about 20 years and 2 months, almost to the date, that I was here at Stanford doing the Summer Research Institute in Geriatric Psychiatry, where I first met Dr. O'Hara, and started my research career looking into late life depression. And that's what I'm going to talk about today in terms of how do we think about mood and cognition in the context of late life depression. Here are my disclosures. All the work that I'm going to be talking about was funded by NIMH. And you just heard from the excellent talk from Dr. O'Hara and Dr. Williams about how cognition plays a really important role in shaping depression treatment outcomes. And when thinking about pharmacological therapies, poor cognition tends to lead to harder to treat depression. But one thing that we don't know is whether cognition also plays a role after treatment is successful. Once somebody has been in remission, does poor cognition have an impact on that depression course? And that's what I'm going to talk about with some preliminary data from the Rembrandt study that I've been doing with Warren Taylor at Vanderbilt and Carmen Andrescu at the University of Pittsburgh, where we're interested in trying to understand predictors of recurrence in late life depression. When you successfully treat older people with depression, about 44% will have a relapse within a 2-year period. And we're trying to understand some of the reasons behind that. And so for this study, we recruit people who are either in remission and follow them for 2 years, or we treat them to the point of remission, and then they enter the main part of the study. The data that I'm going to be talking about is from this initial part of the study where we're treating people to remission. And we wanted to look at features of that acute initial treatment phase to see if that could tell us some clues as to what might be predictors of depression course or recurrence. And so what we wanted to do was to see if we could cluster participants based on things like how long did it take to get better, whether there was cognitive issues at remission, as well as what was the mechanism of action of the antidepressants that they were treated with. And then once we have these clusters, we wanted to see if these clusters could predict outcomes related to recurrence. And then also, are they related to neural markers of recurrence? So one of the points of the Rembrandt study is to use functional imaging, like that stress test that Leanne talked about, to see if there are neural correlates of recurrence that can be used clinically. And so to do this, we looked at 82 of our participants in the initial treatment phase, and all of these participants were treated according to a standard algorithm that was informed by the STAR-D trial and the Duke ENCODE study. And then we used antidepressant mechanism of action information from the psychoactive drug screening program that was developed by Brian Roth at the University of North Carolina. And then to create these clusters, we used a dimensionality reduction technique called ISOMAP. And so we ended up with 3 different clusters, and you can see these clusters in different colors, red, green, and blue shown here. And when you look at time to remission, you can see the ones that spent the longest time getting better are the ones in yellow. The ones in darker colors tended to get better faster. And then we see a similar pattern with the trail making test is what we used for our cognition assessment. The ones that did better are the ones in yellow. The ones that did more poorly are in the darker colors. And then when we look at the dimensions across which these participants were spread, the first dimension seemed to be strongly associated with cognition and time to remission. Likewise, the second cluster was strongly associated with cognition, and the third dimension seemed to be most strongly associated with the antidepressant mechanism of action. And when we look at some of the clinical and demographic features of these clusters, there was no significant difference in these clusters in terms of their age, sex, race, but they did differ based on their baseline depression severity at remission, with cluster 2 having the lowest depression scores compared to clusters 1 and 3. And when we look at time to remission, cluster 1 took the longest to get better compared to clusters 2 and 3. And when we look at cognition in terms of performance on the trail making test, which is a test of executive function and processing speed, cluster 3 did the worst. So cluster 1 took the longest time to get better. Cluster 3 did worse on cognition. And when we look at the antidepressant mechanism of action, we plotted the inhibition constant on the y-axis, and you can see there was a significant difference in the binding affinities of cluster 3s and cluster 1 when looking at alpha-2 adrenergic receptors. So that should ring a bell from what you just heard, as well as the D2 dopaminergic receptor, where cluster 3 had actually lower binding affinities compared to cluster 1. And then when you actually look at what were these patients taking in these different clusters, we look at their modal regimen that they were on. In cluster 1, the modal regimen was a combination of bupropion and escitalopram. And you can see that a lot of the participants in cluster 1 tended to be on polypharmacy, multiple medications at once. And that is a reflection of the fact that they were probably also the hardest to treat and took the longest to get better. Cluster 2 tended to be on monotherapy with escitalopram. And then cluster 3 was tended to be on monotherapy with either bupropion or sertraline. When we look at recurrence risk, how often do people have a relapse? And does that differ according to what cluster you're in? We see a significant difference between clusters 2 and 1. So cluster 2, the folks that got best the fastest with the best cognition had the lowest recurrence rate compared to cluster 1. Cluster 3 was somewhere in the middle. And then another way that we can look at depression course is to look at something called depression load. What is the burden of depressive symptoms over time? And to do that, we basically take the integral of the curve of depression ratings over the time that they were in the study. And as sort of proof of concept, if you look at this depression load statistic according to different categories of participants, either folks that were never depressed, people that were able to stay remitted throughout the 2-year study period, and people that had a relapse. You can see that people that have had a relapse have the highest depression load. And when we look at depression load according to our different clusters, we can see that cluster 2 had significantly lower depression load compared to cluster 1, as well as significantly lower depression load compared to cluster 3. And then one of the things we did was look at neuropsychological performance at baseline and 2 years at the end of the study. And if you remember, cluster 3 had the lowest cognitive performance on the trail making test. And here we're looking on the y-axis at trail making test performance in terms of how long it takes to complete the test. The longer it takes, the worse you're doing. And we can see that there was improvement in cluster 3. Cluster 1s and 2 stayed relatively stable across the 2-year period, but that wasn't significantly, statistically significant. What we did see a statistically significant change was in immediate memory performance. So we did see an improvement in cognition in cluster 3. One of the other things that we looked at were some of the neural markers of recurrence. And as Leanne mentioned, we can use task-based functional imaging as sort of a stress test for the brain. And in our study, we use something called the multi-source interference test, which is basically, you can think of it as a mild cognitive stress test with incongruent conditions and congruent conditions. And these tests activate a whole network of brain regions, both in the cortical space as well as subcortical regions that tend to be stress responsive. And this is just showing a map of all the areas that get activated by this task. And one of the areas that gets activated is the bed nucleus of the stria terminalis. And activation of that region was very important in distinguishing time to relapse. So if you had high activation of this brain region, you were more likely to relapse quicker compared to people that had relatively low activation on this task. And when we looked at this bed nucleus of the stria terminalis activation in our clusters, we found a pattern that was very consistent with what we saw clinically. So cluster 1, right, which is the folks that take a long time to get better, they have the highest depression load, they had the highest activation of this brain region on this task. And then cluster 2, which did the best, had the lowest activation. So that was very consistent with what we saw clinically. So to summarize, we see that fast time to response, good cognition leads to the best clinical outcomes. And that cluster 3 had relatively lower D2 and alpha 2 binding affinity compared to the other clusters, but then had reduced bed nucleus stria terminalis activation and improvements in cognition over the 2-year period. And then cluster 1, they did the worst. They had the worst outcomes, highest activation. And so I think what these preliminary findings indicate is the idea that, what Ruth was saying, that poor cognition might be a malleable target that can actually be addressed pharmacologically. As you saw, cluster 3 had poor cognition, but also lowest affinity for the alpha 2 and D2 receptors. And you heard from Leanne about how guanfacine addresses the alpha 2. And then there's another drug called pramipexole, which is a D2 agonist. And there was a study published just last month in Lancet Psychiatry for patients with treatment-resistant depression, that if you augmented their antidepressant treatment with pramipexole, you had significantly improved clinical outcomes shown here. And so we're excited to target the other receptor that I talked about in the ACE-D study, as Leanne talked about, and you'll learn more about. And with that, I'd like to thank my colleagues on the Rembrandt study, and particularly my former RA, now graduate student, Angie, shown here, who did a lot of this work. Thank you for your attention. That's correct, yes.
Well, we're going to open it up for questions. And so now is really a good time to think of your questions, but I'm going to lead off. Leanne, I'm going to start with you. Do you think that the field of psychiatry is now ready for these more synergized and predictable approaches that both you and Olu have spoken to so effectively today?
Thanks for that great question. Let me check the microphone is on. Yes. I'm very optimistic about, as you may have gathered, I absolutely think we're ready. I actually think it's essential because we need to be collecting data that is relevant to how the information is being used in clinical practice. So we do these studies in a lab-based setting. I think we really need to move into doing trials where we prospectively use the information we're finding so we can really pin down, is it effective when it's deployed to guide or support a treatment decision? So, yes.
And what about individuals where they may not have access to imaging? Is there a future that we can see for them?
I believe so. And I am very inspired by how this has been thought about in other fields. Cardiology is one, oncology, migraine screening, which also uses MRI. And the goal would be to, number one, create more access, which I know the field of radiology is working on proactively, including with mobile systems. Second, to develop what we would think of as surrogates. So you predict from the brain measures what behavior, cognition, digital signatures actually can be predicted from the brain and be used to scale further and monitor and potentially determine when is it the right time for someone to actually come in for a scan.
No, that's exactly the vision that I think we need. Olu, I mean, this was amazing work. Many people don't know that the prevalence of suicide among older adults isn't really significantly different from across the lifespan, but the ratio of attempts to completed suicide is almost 1-to-1. So we have a real risk when people are older. And so your work in really trying to track post-treatment is very important. I'm wondering, does this work necessitate an AI model? You know, we're so good at the fundamental statistics in our field, and where would the value of AI come in?
Yeah, so is this working? I think to Leanne's earlier point, I think thinking about ways that we can have proxy measures of some of the things that we capture with neuroimaging, that there may be a role with AI identifying features for more tractable measurements that would map onto some of the things that we could do with neuroimaging. Because, you know, I was just having this discussion earlier today about whether people are going to actually pay for all of these scans, but there might be cheaper, more accessible formats that are informed by AI.
You know, it's so true and so essential to think of those approaches. Do you think psychiatry as a field is ready for AI approaches? I mean, we've been so trained to hypothesis-driven research. Our diagnostic manual says statistical. We really have some of that excellent training, Helena Kramer and others. You know, it seems often that AI can be characterized as a fishing expedition. Are there approaches we can take to re-educating folks about that?
Yeah, I mean, I think part of it is what was brought up earlier today about sort of the vast amounts of data that we can collect now that I think are really only addressable with tools like AI. So, you know, you're going to hear later today from my colleague, Alex Liao, about, you know, digital phenotyping, and we can capture all kinds of data from all kinds of devices that inform us about our behavior, our cognition, our thoughts, our actions. And so I think, you know, it's important for people to understand that AI, it's like any other thing. It can be used for harm, it could be used for good. And I think educating clinicians about that is really important.
That's terrific. Are there any questions from you? Yes.
So my question, I mean, one of the things I just learned today was about the age, that there is sort of, there are differences sort of in the age spectrum. And talk about late life. I'm someone who's interested in the human potential of the youth, and involved in Stanford with 2 kids who are very different, and executive functioning has been different, both of them. But it was always said that depression is the one that causes cognitive therapy as opposed to the other way. And I was just wondering, this is fantastic work, and I was just wondering, as a proponent of Dr. Williams, what is it that we can do? Because it's obvious that cognitive therapy should be just done in school, like that should be sort of, instead of just studying math and statistics and whatever else. Cognitive training seems to be a precursor for mental health all throughout the life. So you know what I mean? I'm also curious about AI, whether it's agentic AI or just AI, to help maybe, because the youth are ready. They don't want their human potential to be wasted, but they're not able to sort of go to that next step.
So Leanne, maybe to you first, and then Olu.
You always ask fantastic questions. So what can we do for I agree, the younger generation is ready and understands mental health. So in my mind, it's a suite of things. One is fundamentally incorporating mental health into our education, because we learn from when we're very young about road safety, general health, and we kind of scared, I think, to just say mental health is part of all of us. It's our brain is our biggest asset. Let's learn about how it functions and when might it be disrupted, and just have that actually part of the education. That, I think, leads naturally to the kinds of solutions that you're talking about. Same as we have so many apps and training programs to help us get physically fit. We can then think about, well, it's a natural, it's part of our health, our brain health, to have all those available. And to understand your point about, we have now learned that poor cognition, which is so important to how we function, is not a consequence of mental illness. So we really need to pay attention to it. There are so many concrete ways we can answer it. I wonder if Olu's got some more.
Well, I was just thinking about, I have a 9-year-old daughter, and I've been struck by how many things that I didn't learn about until I was in residency that they're learning early on in elementary school, right? There are books for kids on mindfulness, right? My daughter's school now, they have a digital wellness curriculum as part of it. So we're seeing, I think, more of these approaches being done and addressing a lot of the issues that you're talking about.
Anyone else? Yes, right here.
Dr. Williams, you talked about the different biotypes, but one thing is overlap between the biotypes. Again, I think one of the things you did highlight is precision in terms of which part of the brain these biotypes originate. But are there, again, when you characterize them, is there overlap between the biotypes, and how do you then direct them towards a specific biotype for treatment?
Such a great question. Thank you for asking it. And this is about, do the biotypes that I talk about overlap? The ones that I focused on, I purposely focused on the brain circuits that are independent of each other as a starting point. So results in, we can actually classify independently biotypes for 90% of the people with depression that we've looked at. And this is a very pragmatic approach. So we are seeing, here are 6 brain circuits that we've already found are independent sources of information. And so we have shown through our machine learning models that each 90% of people have that primary dysfunction that's most notable. It's the highest or the lowest. Many people will have a second dysfunction, but it usually will be of lower magnitude. And this is where the next step is to say, how can we now refine this and explore further? It's unlikely it's going to continue to be nicely, neatly separated. So we want to really do that deep dive into the bigger data sets with more powerful models and really look at how can we expand them and really work out where the boundaries are, and are there combinations of circuits that are important in some cases.
And that's probably where the value of AI approaches will become particularly important. And also, we talked today a lot about brain and cognition, certainly including cognition, as we did in the exposure therapy case. Not only was it the circuit that was involved, but then adding performance objectively on attentional measures actually added significantly to our understanding of a group of individuals who did not respond. So those are the refinements. And just as if the story isn't complex enough, we haven't talked about stress, sleep, so many of the other physiological systems that may also be particularly valuable for actually carving out domain areas where, in fact, people don't exhibit the best treatment response. So it's really, and this is particularly evident in geriatric psychiatry, I believe, Olu, because when you talk about polypharmacy, it's the nature of the beast as we all get older. Sure. Yes, so I think we have here and then there. Yeah, I should. Yes, I'm being very hemispheric specific in my question.
So obviously we know that depression and mental health as a whole is very heterogeneous. So I'm very curious about how you see the biotyping in the diagnostics process. Do you think in 10 years we will laugh at like the way we currently diagnose mental health and it will be completely redefined in that way?
Yeah, I can address that. I think it's a real problem in our field that when we diagnose somebody with depression, it's equivalent to diagnosing somebody with a fever, right? We don't know what the cause is, but with Leanne's approach, we're getting to a more etiologic, pathophysiological way of defining diagnoses that will lead to more specific treatments, right? And so. I think we're laughing at how we diagnose things now, but I think with the work that we're all doing collectively, we're working on a new version of the DSM that will hopefully be out in 3 years. And there's a lot of discussion about how do we incorporate the kind of work that Leanne is doing into the next version of the DSM? How do we create a pathway for biomarkers to be a part of our diagnostic process? I think that's really important.
On this side, and then I think we have a question at the back there too.
This one is
Excellent talks. Thank you all so much. I was intrigued to hear about the ways that we can use the depression biotypes for intervention, but I'm also very curious about how they could be used for prevention. So has there been any research into where these circuit dysfunctions manifest neurodevelopmentally or when they manifest relative to clinical symptoms?
There's, I don't think as yet as applied in terms of using Leanne's models at an earlier developmental stage. And I think, but there is definite interest. And there are a lot of data sets where, in fact, imaging has occurred where we could look at the various biotype data and circuits to glean more. We know in childhood disorders, there's a lot of interest, particularly in disorders that also have cognition at their base, like autism, for example. There are probably good extant data sets that could be examined to look specifically at these questions. And it relates to Nusha's point earlier that should we be capturing this earlier in the lifespan and sort of have cognitive exercising or enhancement of these particular biotypes right from the get-go. That I think would be the ultimate dream. We had a question and then I think we maybe, yes.
One in the back.
Yeah, that's where, one in the back over there.
Here.
Yeah.
And one over here. Then one.
There you go.
Okay. Hi, thank you for the talk. You mentioned one of the primary goals is really to develop this personalized treatment plan and reduce the trial and error approach and how there's more precision in other aspects of medicine. But oftentimes, even in other parts of medicine, there is a trial and error approach before you get to the imaging. For example, with migraines, you have to take all the different standard medicines. If that doesn't work, then you lead to imaging. You have an injury, you start with physical therapy. If that doesn't work, you go into imaging. So how do you see your research or just generally that approach changing specifically within mental health?
Yes, you.
It's a great question. And that's part of my emphasis on saying these trials need to be done in the real world setting so they mirror the decisions. I think the main thing for mental health is to reduce the really long period of trial and error, because depression 7 years in general in the field, it's 11. So I would anticipate, and I'm sure my clinical colleagues would say there would need to be that similar approach following clinical guidelines, but just to shorten that time to selecting.
And you said we have one last question. Right there.
Hi, I appreciate all your work. I'm not going to worry about it. I wondered if there's studies done on the consistency of looking at a functional MRI and diagnosing a biotype over time with treatment or without treatment, because my postulation is that the brain changes. And how can you label somebody kind of like with a photograph of a functional MRI at one point and then not recognize that it changes with, social determinants of health or other treatments, and label that person?
Definitely to you, Lee.
It's a great question. So maybe we go back to the cardiology analogy of you would have a system, let's say blood pressure, too high, too low, you're trying to get back in the healthy range. So the circuits that we're measuring in our case, they can be reliably measured over time. What does change and we want to change is when that circuit is outside of the healthy range. So we can identify, is there, and we're calling it a biotype, which implies fixed, but we're really meaning what is the problem that we're identifying. So if it's too high, the measurement is stable, but we can track that, we can restore it to the healthy range.
Which is the value of Leanne's continual assessment of how other fields are approaching this in medicine, because so often we're a little more reticent in mental health. And one last question that has been trying to get in, right? Yeah.
Sure, thank you. My question is actually sourced from having a loved one who passed 25 years ago, the brain condition. That's okay. So I have a question about a cheaper test. Do the 6 biotypes you have identified have any chemical or excretion trail? So obviously a blood test or a swab is much cheaper. So I'm just wondering, has the chemical aspect been investigated?
Olu, I think we bring that to you.
Yeah, there's actually, I participated in a symposium yesterday, and I believe Steve Hyman is part of this huge psychiatric biomarkers network where they are focused on more fluid-based, whether it's CSF, blood, type of biomarkers for different psychiatric conditions. So there is work on that. Now, how that maps onto Leanne's biotypes, I'm not sure, but that work is happening sort of in parallel.
Maybe I take the one challenge we have is exactly the issue you highlighted about this focus on the cost of imaging, and that's something I really think is worth revisiting. We spend a lot of time working with radiology and healthcare providers, and the modeling, the cost is manageable when you look at the equivalent cost separate from, say, a facility's cost, and you compare it to what you save if you're getting someone well in, less than 7 years. Let's say we can get them well within a few weeks. So the modeling, I think, it hasn't been done in a clear way yet for our field, but I think it's a really important case to make because we benefit from existing workflows that exist for every other area of medicine. whereas we absolutely need to look at blood-based markers, all sorts, is how to build those clinical pathways in our field. Yeah. So thank you
Terrific.
for your question.
So I think that's probably it, so we don't go over. Thank you to everyone. And don't hesitate to reach out to us all during the symposium if you have additional questions. Thanks so much.
Thank you for such an engaging and thoughtful discussion. We'll now take a break and reconvene at 10:50. So please.